Welcome back, my microbiology students. Today we're going to go over chapter 13, which is a continuation of last chapter. In chapter 12, we talked about the host defenses that incorporated the innate immune system, which was essentially the first and second lines of defense. And in this chapter, we're going to cover the adaptive immunity arm, which will cover the third line of defense. So here we have at first a couple of learning outcomes for the section. We're in 13.1 right now. The 13.1 section, again, is going to give us an introduction to our adaptive immunity. That is our third and final line of defense that will come into play if a pathogen makes it past the first or second line of defense. So it is activated when the other two fail to get rid of the pathogen. The adaptive immunity is only acquired after an immunizing event, such as infection, which means exposure of our adaptive immune system to some sort of pathogen. The cells that are a large part of this process are B and T lymphocytes. So other things that we talked about that were cellular were in the second line of defense. So make sure that we're clear on which types of cells come into play in which line of defense. So the third line of defense, again, are B cells and T cells. And those are two varieties of lymphocytes that will undergo selective processes that will prepare them for reacting to specific antigens or immunogens. And with that, we have this term that we need to familiarize ourselves with, which is immunocompetence. And immunocompetence is just the ability of the body to react to foreign substances. So whenever you have a patient, for example, who does not have a competent immune system, that means their immune system is weakened. They don't have a strong immune system to react to foreign substances. So immunocompetence is the ability of our body to do that. So a couple of terms we also need to be familiar with for this chapter is antigens and immunogens. So antigens, by definition, are molecules that can be seen and identified by the immune system. And most antigens, but not all, many antigens, at least from foreign substances, are also considered immunogens because they provoke a immune response. So not all antigens will provoke an immune response, but any of them that are considered immunogens will. And to give you an example of the difference there, so on our red blood cells, I know that you in A&P are probably very familiar with this. My biology students we might have talked about this a little bit, but we talked about how red blood cells have proteins on them that we call antigens because they can be detected by the immune system. So if this person here has the type A blood type, they will have type A antigens, and these antigens are proteins. And our immune system identifies those A antigens as self. So they do not provoke an immune response. They're still antigens because we can identify them, but they don't provide an immune response or a reaction, so they're not considered immunogens in this case. However, if we were to transfer blood from a donor that is type A into a host that is type B, that would not be good because this person, the type B person, would then recognize those antigens as foreign, and they would be considered immunogens in that case. Their immune system would recognize those A antigens and they would attack them, and it would essentially, within 10 milliliters, would cause death for that patient. So that would mount a severe immune system transfusion reaction. Alright, so that's the difference between antigen and immunogens. Immunogens are antigens that provoke an immune response. Antigens is just anything that our body can detect as self or non-self. Any exposed or released protein or polysaccharide is potentially an antigen, even those of our own cells. Just like the example I gave you with our blood cells. Our body recognizes those proteins on all our other cells, as well as polysaccharides, which are carbohydrates, as self. But if it recognizes it as non-self, then it attacks. And so our own antigens do not typically evoke a response. Some additional characteristics and terms we should know when it comes to adaptive immunity is that it is specific. So unlike the innate immunity that we described in our last chapter, we see specificity in this chapter. So what I mean by that is in last chapter we talked about things that would protect us in a non-specific way. For example, a unbroken skin protects pathogens of any type from getting into our bodies. Our tears, the mucous membranes, certain sequences of or complexes of carbohydrates and proteins like the PAMPs and RAMPs that we talked about in our last chapter, that's very generic. Here what we're seeing is we're going to attack specific antigens by making antibodies. And so that's something new we're seeing here, antibodies. We did not talk about antibodies in chapter 12 because it is specific to the adaptive immune system. So whenever we say specificity, these antibodies will be produced to the specific antigen, such as the chickenpox virus. And the same antibody that's produced against the chickenpox virus will not work against a different virus like the measles. So it's specific to a specific pathogen. And in this type of immunity, the adaptive immune system also generates memory. And memory means that our immune system essentially remembers something that it's come in contact with before. They have the ability to recall their first engagement with some kind of pathogen, and it helps them to create a much more intense response the second time that it sees that pathogen again. Alright, so next in this section we're going to describe the stages of immunological development and how these cells interact with pathogens. So in brief, again, we're going to describe mostly interactions and development of B and T cells because those are the ones that are involved in this type of immunity. So all lymphocytes, regardless if they're B or T lymphocytes, arise from the same basic stem cell, and that cell is called a hematopoietic stem cell. It actually also is the precursor cell to the other blood cells that we've talked about prior. But it's located in the bone marrow, as we've talked about before. Now, once these cells have proliferated and created B cells and T cells, or cells at least that will be destined to become those cell types, they'll mature in different areas depending on what cell that they will become. So if we have a immature lymphocyte and it travels, or it doesn't travel, excuse me, it stays in the bone marrow, then it will become a B cell. But these immature cells that travel to the thymus will become T cells. So again, we start off with our hematopoietic stem cells in the bone marrow. Some cells will stay in the bone marrow and become B cells, and others will travel to the thymus to become T cells. Alright, so both of these, after they have matured in the thymus or the bone marrow, will then go on to move to other lymphoid organs. And then after that, they will constantly circulate throughout the circulatory system and the lymphatic system, working as a surveillance mechanism for any potential pathogens that is in our cellular environment. Alright, so next we have a picture of our text that also is describing the same thing. It's showing you the overview of that lymphocyte development. Again, we have our precursor cell, which is the stem cell. Right, let's see here, stem cell, and it's in the bone marrow. I just added in the name, which was hematopoietic stem cell, and again, it's in the bone marrow. Some cells will stay in the bone marrow. As you can see, this red stuff here for y'all biology people, that's showing you bone marrow. AMP, I'm sure y'all already knew that. But these are destined to become B cells. So the ones that stay are B cells, and think of it this way, B for bone marrow, B cells. All cells start in the bone marrow as far as blood cells, but the B cells that are B lymphocytes stay in the bone marrow. Oppositely, if any of these immature cells travel to the thymus, they will become T cells. So again, thymus T cells, T for thymus, T for T cells, bone marrow, B cells. Now regardless, once they're mature, either in the thymus or the bone marrow, they will travel to the lymphatic organs such as the lymph nodes. We have the spleen here as well. Then after that, they will wind up coming in contact with various pathogens potentially and elicit different responses that can vary depending on the cell type that we're initiating with, either the B cell or T cell line. We'll get into some more of these specifics later. So, when pathogens are carrying antigens across the first line of defense, at that point, any kind of phagocytes that are existing in that area will migrate to the site. Our tissue macrophages will ingest that pathogen, and it will initiate an inflammatory response by releasing certain chemical signals. Other cells like dendritic cells can ingest the antigen and migrate to a lymphoid organ, such as the lymph nodes or spleen, and they can then present that antigen to a T or B cell. So again, looking at the big picture here, let's say we have a foot. Oh my gosh, I probably should not draw this because I'm not good at using this thing. Here's the, okay, we'll say it's a hand. It looks more like a hand. And let's say we get an injury, all right? Whoop, bleeding, we have an injury. And it's because we got a rusty nail somehow in our hand. Well, that site of injury is going to elicit these chemical signals that will attract those phagocytes. And again, phagocytes are cell eaters, meaning they are cells that eat stuff, debris and pathogens, as well these macrophages, which are a type of phagocyte. They will ingest the pathogen along with dendritic cells in the area, and then these collectively can send these cells that have ingested it to a lymphoid organ like a lymph node, and they literally will take a piece of that pathogen and show it. They'll present it to T and B cells. So when we say that it's being shown the pathogen, let's go into a little bit more on exactly what we mean by that. This is where we're going to get into conversations about cell markers and cell receptors and cell communication. So all cells have a variety of different markers on their surfaces. And when we say markers, we're talking about things that can be recognized, again, antigens essentially. And these markers can be utilized for detection, recognition, cell communication, and they play a very important role in the different components of immunity. Let's look and see some examples. For example, we have our MHC complexes. That stands for Major Histocompatibility Complex, and it's a set of genes that code for cell markers or receptors in humans that give rise to various glycoproteins that are found on all cells that have a nucleus, which is all cells except red blood cells. Some textbooks will call this, including ours, it'll use both terms, the HLA system, but it's basically a description of the types of MHC genes that you have. But these markers play a vital role in recognition of self by the immune system and in rejection of foreign tissues or cells. Now, I'm going to show you all the three classes of genes. Just make sure that you notice I have written here in the highlighted area. You don't need to memorize these definitions here in the different classes, but you should recognize that there are different classes and that they code for these MHC markers and receptors and is used in this self and non-self identification. So we have three different classes, class 1, 2, and 3. Our class 1 genes, they code for any of the markers that are going to appear on nucleated cells in regards to our HLA system. And our class 2, they're going to be important regulatory markers. They're found on various cell types that are involved in presenting antigens to T cells during immune responses. And class 3 genes, they encode proteins in the complement system, among others. So again, like I said, you don't need to memorize these three definitions I'm not going to ask you to differentiate between the three on the exam, but do know what they're used for. They are markers, protein receptors that are used in self and non-self ID. So let's look at some examples in picture form. Here what we see is a class 1 MHC, and again these are found on all nucleated cells, which is all of our cells except for red blood cells, and we see the plasma membrane here and our MHC compatibility complex embedded. In our class 2 MHC, we have those are going to be found on some types of white blood cells. Of course, there's also the type 1 because we said they're found on all cells except red cells, but these specific cells, such as the white blood cells, will have this additional class 2 MHC complex marker found in its plasma membrane. Alright, let's talk about some other markers. So we talked about MHC markers. Let's talk about CD markers. CD markers stands for cluster of differentiation, and they're called that because there's about 400 of them that have been named so far, and they're oftentimes going to have a specific pattern of them depending on cell type. In fact, certain cell types only display certain of these markers. So some of the most important ones, as we've seen listed below, are CD3, CD4, and CD8. And just to give you an example of how they can be they can serve as an identification for the cell type, is that CD4 and 8 are found on T cells, but specifically CD4 is found on T helper cells, and CDH is found on our cytotoxic T cells, and we'll get into more on that a little bit later in the PowerPoint. But again, they are proteins that are embedded into the membrane of the cell that is utilized as part of our immune response. So what's the role of these receptors, these MHC receptors and these CD markers? Their major role is to accept antigens in some type of way. B cells have receptors that bind antigens. T cells have receptors that bind antigens. With T cells, though, they typically have to have been processed with the MHC molecules and presented to the cell surface, and there are potentially billions of unique different antigens. So when our immune system is challenged with an antigen, both B and T cells are going to proliferate and differentiate. Proliferate means to make more. So essentially that's the process of mitosis, which everybody should be familiar with. And then they also have the ability to differentiate, which I think everybody should know what that means. But if not, differentiating means that a cell becomes a specific type. So a clone, another term we're going to learn about, is a group of genetically identical cells is created. That's the proliferation part. All right, so proliferate via mitosis many times will create many clones of genetically identical cells, which y'all should have covered in A&P and biology. And then some clones become memory cells. And those memory cells will serve that memory function of the immune system that will allow us to, quote unquote, recall an infection after having experienced it once before. Now, they both proliferate and then differentiate, but their responses are going to be different. So let's look at how T cells do it first, and then we'll go into B cells. So when a T cell is challenged with an antigen, they will react differently based on their type. There are 3 main types of T cells. There are others in the text, but these are the ones I want you to know. We've got our T helper cells. Oftentimes we'll see that abbreviated as T sub H. These are going to activate macrophages. They'll assist B cells, and they will help activate our cytotoxic T cells, which brings us to the third one on this list, our cytotoxic T cells. These are responsible for just destruction of the infected host cells or any other foreign cells, like a protist, for example. You can think of the cytotoxic T cells as like the infantry here. They are the ones on the front lines doing the actual fighting and the destruction. The helper cells, on the other hand, more are the helpers. They're assisting all of these other different cell types to do what they need to do. And then we have our regulatory T cells, which is typically going to be abbreviated as TR, and this one's TC, by the way, but TR, and they control the T cell response by secreting anti-inflammatory cytokines or preventing proliferation. Because remember, all these other responses are kind of upregulating the immune response, making cytokines, making inflammatory things, making lots of cells, but we don't want our immune system to get out of control. And that's where these regulatory T cells will come into play by down-regulating some of these processes. If we have too many cytokines that are released, we'll have too much inflammation, and that can actually be a bad thing. So we want to keep our immune system response under control. Just like everything else in the body, we have homeostatic mechanisms to regulate everything from temperature. I would have talked about in my class in biology, as well as AMP. And let's see, I know in my classes we've talked about insulin regulation. I bet you talked about that in AMP too. But just like everything that y'all discussed that have homeostatic responses, here we're seeing that as well with these T regulatory cells. Now, one key thing to remember though, make sure you, whenever you think antibodies, think B cells, because our T cells do secrete cytokines that can assist in inflammation and destruction of pathogens, but they don't produce those antibodies. That's specific to B cells. So now let's look at B cells. Speaking of, when a B cell is activated, it divides and differentiates. And some of those cells that it differentiates to are called plasma cells, and these plasma cells are the ones that release antibodies into the tissue and blood. And those antibodies will attach to the antigen that they're specific for, and it will be marked for destruction by the immune system. Alright, and I will skip this slide. We're going to go on to 13.2. I changed my mind. We'll go back to this slide and do it. So this is just a little concept check to make sure we're kind of all on the same page. But B cells mature in the blank and T cells mature in the blank, and then both cells then migrate to the blank and blank. So if we'll recall, we said that all of the blood cells start in the bone marrow via hematopoietic stem cells, but we said cells that become B cells will stay in the bone marrow and become B cells. But if it's a T cell, it will mature in the thymus. Both cells will then migrate to other lymphoid organs, such as the lymph nodes. I'm going to write LN because I know I'm going to run out of space, and the spleen. So that will leave us then with the answer being D. Next, we're going to move into 3.2, which will help us to summarize the processes that are going to go on with B and T cells throughout their maturation and throughout the, we'll talk about the structures that they have and how clonal selection and deletion is completed. So we'll start with T cells first. As we've described before, we said the maturation of T cells and the development of the receptors are going to be directed by the thymus gland because that's where they mature. They're going to contain a few of those CD markers that we mentioned prior. Same ones. Remember, there's over 400, but I only want you to know these three. The CD3, 4, and 8. These CD3 receptors are going to surround the T cells receptors and assist in binding. They're basically like co-receptors. They're just little assistants. CD4 receptors are going to be the type of receptor that helps a T cell bind to class 2 MHC molecules. And then CD8 are going to be found on our cytotoxic T cells, and they bind to the MH class 1 molecules and basically mark them for destruction. So let's look at some pictures so we can get a visualization of this. Again, we have a T cell shown here from the thymus, and if we blow up this cell really big, it's showing you the plasma membrane all around. We see a CD3 marker, we see the CD4 marker, we see our MHC class 1 um, complex, and then we see this antigen binding site. And so utilizing these CD3, CD4 markers, as well as the MHC complex, it's going to attach to some kind of antigen that it can detect. Let me also say too, I want us just to be reminded really quick, since this is a CD4 marker, this is going to be a T helper cell. This could have been a CD8 marker, and if that were the case, it would have been a cytotoxic T cell. So now let's look at our specific events that occur in B cell development. And again, we said that this is going to occur in the bone marrow. And so initially they are what we consider naive B cells, and they are going to be considered naive because they have not come into contact with an antigen that is foreign yet. Essentially, during this developmental phase, the naive B cells are going to circulate the blood and travel to different areas and just learn basically what is self. So if we look at the cell surface of B cells, we will see we still have our MHC class 1 molecule, but also our MHC class 2 markers. And we also have, in addition to that, antigen binding sites in the form of antibodies. So antibodies are also, by the way, called immunoglobulins. You'll see both of those terms throughout the chapter. So how do these cells build immunological diversity? And what we mean by that is, how do they begin to establish a way to determine self from non-self in so many different unique antigens. So by the time B and T cells reach our lymphoid tissues, they are equipped to respond to a single unique antigen, every single one of them. And that diversity is generated by rearrangement of gene segments that code for antigen receptors that are found on those T and B cells. And with those rearrangements, every possible recombination can occur. So basically then we have this huge assortment of different B and T cells, and each one of them can recognize a unique thing, a unique antigen that is not self. So it's estimated that the human antibodies produce, that each human produces antibodies with about 10 trillion different specificities. Pretty amazing if you ask me. Now, you are not responsible for knowing the mechanism behind all of that variability. That's a very technical, more of a molecular biology thing, so I'm not going to hold you responsible for it. But essentially, if we look at the structure of an antibody, it looks like, generically anyway, they look like Ys. And so these little Y-shaped structures are made up of different chains. We have the light chains here and here, and we have the heavy chains, which are the longer ones on the inside. The top half, which includes the light chain and the first part of the heavy chains, is called the variable region of the antibody, and the other parts are called the constant region. And you could probably guess that's because among antibodies, this region here at the bottom is constant. It's typically the same for each one. But here at the ends, which is where antigen will bind to, is very variable. So whenever we have this genetic shuffling while creating the antibodies from our genetic components, and as hopefully maybe we'll recall, DNA is converted to RNA, which is converted into protein, and these immunoglobulins, these antibodies, are proteins. So these proteins are made up of various recombinations of RNA transcripts that we derive from the same gene products in DNA. Again, you're not responsible for memorizing that mechanism, but for those who are interested, that's how we get all of these different varieties of antigen receptors on these antibodies. So in this, for those of y'all who took biology, maybe you'll remember when we talked about alternative splicing in genes. So that is related to this process, how we can have one gene but get many different proteins from it. In this case, the protein's an antibody, and it's specific to the variable region on the antibodies. All right, so again, antigens are also called immunoglobulins. Oftentimes they are abbreviated as Igs, and we'll go into a little bit more details on different types of immunoglobulins momentarily. But to review a little bit more about the structures on our antibodies, these antibodies are large glycoproteins, and they serve as antigen receptors for B cells. Remember, they're found on B cells. My drawing's terrible, I know. As well as the MHC class 1 and 2 complexes. I'm not going to draw those because, as y'all can see, I can't draw. But they also can be secreted. So these antibodies are going to be embedded into the plasma membrane of B cells, but they're also excreted by B cells. So again, we talked about the light and the heavy chains, the variable and the constant regions. I don't expect you to know heavy and light chains, but I do expect you to know that this is the variable region and this is the constant region, the other parts. And the variable region is the antigen binding site. And I hope that makes sense, because again, we said that this variable region is creating lots of different varieties of antibodies that will bind to different things. And these things are antigens that are found on potentially pathogenic substances that enter into the body. All right, so let's look at next T cell receptors. So in comparison, again, this is a B cell receptor. It looks like generically like a Y. For you AMP people, you may already know that it's not always looked like a Y, but for now, let's just keep it simple. But these T cell receptors belong to the same protein family as our B cell receptors. They're similar to B cell receptors. They're still going to be formed by genetic recombination events that yield variety and constant regions, as you can see here in our T cell receptor. The end is going to be the variable region, and then we have a constant region. They're inserted into the plasma membrane just like the B cell ones were, and they have an antigen binding site right here at the top, just like our B cell antigen binding sites were at the top in that variable region. However, they're relatively small compared to the B cell Y-shaped receptors, and they're never secreted. So the only time we see secretion of antibodies is in our B cells. All right, so next, let's talk about clonal selection. This is the mechanism by which the correct B or T cell is activated by any incoming antigen. So we said previously that our specificity for lymphocytes are already pre-programmed, and that exists in our genetic makeup before the antigen is ever entered into the tissues. So we have essentially antibodies on our B cells, or the CD4 the T cell markers that are specific to pretty much anything that is non-self that our body can come in contact with. So we have those prior to becoming infected with, I don't know, Staph aureus or whatever the case is. So those exist due to this genetic reshuffling to make such a variety of things that we can detect. When you have trillions of things you can detect, you're probably going to be able to detect things that exist. So each of these genetically distinct lymphocytes expresses only one specific, single specificity, and it can react to only that chemical epitope, which is another term that we're going to cover now. Epitope is the region on an antigen that an antibody detects. So here we see our antibody. This is going to be one from a B cell because it looks like a Y. And then we said that this is like the constant region. This would be the variable region. And if we compare one antibody to the next, those variable regions are going to be different for every single cell. So whenever we come in contact with a pathogen has lots of antigens on it. This would be the pathogen, the bacteria cell. And do y'all see all the little green thingies that are surrounding it? Those are various proteins embedded into the plasma membrane of that bacteria cell, and there are certain parts or pieces of those membranous proteins that will bind with these antibodies. This is showing you, hypothetically, let's say that this protein is that one. And so it's just kind of showing you a really, really close up, and we see, ooh, it has some bumps on it. Those bumps are the specific site on that antigen that this antibody will recognize. It will bind to this antigen binding site on the end, and that is the region it will bind with, the epitope. The epitope is the specific part of the antigen which the antibody will bind. So then what happens? We'll get to that in a second. Before we do, let's talk about clonal deletion. If you think about it, whenever we have this huge variety of different things that the body can detect as foreign, due to that random reshuffling of our genes that create that variable region, there is one potentially problematic outcome. Because it's random, we may incidentally lead to lymphocytes that have antibodies or receptors that would bind to self antigens, and that would not be good. We don't want our body to recognize self as foreign, because then it would damage those cells. So there is a deletion process, a clonal deletion process, by which any of these clones are going to be destroyed in development. So if we look here, we see this lymphocyte stem cell, excuse me, and then as part of that replication process, making lots of clones, we'll see that each of these are going to have slightly different receptor sites, but some of them, like this one and that one, recognize self proteins. So we're going to eliminate those. We're going to get rid of them. And so again, this is called clonal deletion, because we're eliminating the ones that are going to recognize self. So what we have left after clonal deletion is the ones that don't recognize self. So if you compare the different cells here, you'll see it's the same ones here. We have the little yellow bent Y. We have the field goal looking one. We have the flag looking one. We don't have this one because it recognizes self. We don't have this one because it recognizes self. But we have this one, and we have the green arrow. So all of those are representing lymphocytes that have various receptor sites, and these now have no possible ones that recognize self, because those were deleted in the clonal deletion process. So now what will happen is that we have a population of these lymphocytes that don't recognize self, that are specific to anything non-self. And then when we come into contact with a pathogen, and again I'll draw a little bacteria cell over here, come in contact with a pathogen, it's going to have antigens on its surface, and those antigens are shown here. Remember, they have epitopes, or specific regions on them that is detected by the immune system. And just by that random chance of creating all possibilities of foreign content, this cell has a receptor site that fits that epitope. And so then we will have a clonal selection process, where we're going to mass proliferate a lot of those cells to mount an immune response quickly for that specific epitope. Alright, so next, moving into 13.3, we're going to list some characteristics of antigens that optimize how immunogenistic they can be. We're going to describe how our immune system responds to allergens, superantigens, and alloantigens, and we'll describe the types of cells that can act as APCs, or antigen-presenting cells. So, in our second stage of the process of responding to antigen, we will see that they are going to be presented by antigen presenting cells. So in order to be perceived as a immunogen, something we should attack, it has to meet certain requirements though. It has to be foreign, it has to have a shape, a size, and accessibility. So some good characteristics of immunogens that provoke strong immune responses are any kind of something that is a complex composition. Proteins are excellent in mounting immune responses because they are highly complex. Their context, which means what types of cytokines are present where the pathogen or a potentially immunogenic thing is, and their size. Typically larger things are better than smaller things, but that alone is not going to be sufficient enough just because it's small for it to be antigenetic or non. So it's just a piece to the puzzle. So this shows you some examples of good and poor immunogens, excuse me. So again, proteins are great immunogens. Microbial cells and viruses, bacteria. This looks like a yeast cell to me, budding yeast, virus. That looks like a spirochete or another spiral-shaped bacteria. We see plant molecules. They can be great forms of immunogens. Human cells, other animal cells, if we were to inject, just like we talked about earlier, the wrong blood type and transfuse it, it would lead to devastating outcomes. If we took cells from another species and just inject them into our bloodstream, that would not be good. They are very good immunogens, and our immune system will respond. Some poor immunogens would be more simple things, like here we just have a simple trisaccharide, which is a chain of 3 sugars, and then we see just a linear polypeptide chain. It doesn't have any shape to it, so it's not very complex. So some good antigens fall into these categories typically. Proteins, we've mentioned those because they are just excellent. They're typically very complex and have 3-dimensional shapes, but these proteins can be in the form of enzymes, cell surface proteins, and even as toxins. Certain bacteria produce toxins that our body can detect. Lipoproteins, which is found in cell membranes, for example, we talked about gram-negative and gram-positive cell membrane differences before. Glycoproteins, which are blood cell markers, the ABO group, for example, any kind of nucleoproteins that are DNA that's complex to proteins, polysaccharides, liposaccharides, again in bacteria cells, for those two and other things. But all of those are great examples of antigens that our body can recognize and therefore are immunogens as well. So what do these APCs do? In most immune reactions, the antigen must be presented to lymphocytes by an APC. And an APC stands for an antigen presenting cell. And these APCs can be B cells, dendritic cells, or macrophages. So what we have here is this here is showing you an APC, antigen presenting cell, and we can see it's engulfing a microbe. So let's say this is it could be a macrophage or B cell, dendritic cell, but it's going to engulf that thing that it's detecting as foreign, and it's going to chew it up essentially. Remember, our macrophages and other cells like it, our phagocytes, they have digestive capabilities. They're going to then take a piece of the bacteria or virus, whatever it is, this microbe, this pathogen, and present it using the MH2 receptors. So here we see the MH2 receptors that are naturally found on our APCs, and we're seeing these little pink things now. These pink things are immunogens that were found on this microbe, and it's going to present them by binding them to their MHC2 receptor, and then it's going to show them to another type of cell, such as a T helper cell, which is what we're seeing in this picture. We're seeing the same. We'll say it's a macrophage here that has engulfed a pathogen, and now it is displaying a piece of it, the immunogen part, the specific epitope of that immunogen, onto its cell membrane via the MHC2 class complex. Then a T cell, like this T helper cell, can go over to it and it can bind to the T cell receptor in addition with the CD4 receptor as an assistant. Remember we said the CD4 and CD8 and CD3 CD markers can ultimately, they work together depending on the cell type to serve the same function. They're going to make it to where they can detect a antigen from an APC. All right, so now let's move to 13.4. We'll describe the main differences between T helper and cytotoxic T cells. We'll explain how native T cells become sensitized to an antigen, and we'll look at similarities and differences between some of the other T cell types. Excuse me. All right, so in stage 3 and 4, our T cell reactions are going to be the most complex and diverse in the immune system and involve several subsets of T cells. So let's review for a moment. We said an APC, the antigen presenting cell, is going to engulf a pathogen. It is then going to present it to that T cell by displaying it on its MHC class 2 molecules. The T cell is then going to receive that epitope that it is being shown by the APC, and then lots of things happen. And we're not going to go into all the details, but to make things simple, the end result of when that T cell is stimulation is the mobilization of other immune cells. That can include phagocytes, B cells, other T cells. There's various subsets of T cells, but again, the only ones I care that you know about are on the PowerPoint. So, such as our CD4 T helper cells, these are going to have the CD4 marker on them. Their job is to activate other immune cells. The cytotoxic T cells, they are going to directly destroy cells, particularly ones that are virally infected or cancerous or are marked as not human cells. Our gamma delta T cells respond to certain kinds of PAMPs in the same way as our nonspecific white blood cells did in the innate immune system from chapter 12. There is a benefit of those gamma delta cells. They do respond more quickly. They do produce memory, which is not like what we saw in the innate immune system last week. So for that reason, they're almost considered like this bridge between the innate and the adaptive immune responses. Moving to 13.5, we'll diagram B cell activation. We'll look at another drawing of antibody molecules. We'll explain the various end results of what happens when B cells are stimulated. We'll talk about types of antibodies, and we will also talk about primary and secondary immune responses. So this is basically a reiteration of the concepts we talked about previously with our shape of our antibodies and their structure. They have a symmetrical Y-shaped arrangement, which again I'm going to say quote unquote because that's not always necessarily true, but generically they are shown in textbooks as Ys. And they have antigen binding fragments, which is in the variable region. They have a constant regions down here. And then this variable region, or that where the antigen binding site is the location where the epitope of an antigen will bind and is found on the surface of B cells, also excreted by B cells. And specifically, those B cells are called plasma cells. Alright, so let's look at how B cells are activated. So initially we're starting off with a microbe here, and it will come in contact with a B cell, and that B cell will bind to the epitope that its receptor fits, or alternatively an APC can present it to a B cell. Just depends on, kind of, I guess, which came first in contact with. But here we have the microbe, it's showing the B cell, we see the B cells engulfing it, and it's now going to display a portion of that pathogen on its cell membrane via the MHC class II molecules. It is now going to show it to a T cell. That's why it's called an antigen presenting cell. It's presenting the antigen to the T cell. And this is showing that very big. So we can see the MHC class II molecule. We see our CD3, CD4 T cell here, which is a T helper cell, and it is showing it the antigen that it has come in contact with. So what happens after that? That activates the B cell. The B cell then will begin differentiation and clonal expansion. Differentiation, again, is the habit of creating more cells that become more specific. So this precursor cell is going to undergo mitosis, 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 and some of the subsets of those cells will become regulatory B cells. Some of them will become memory B cells, and some of them will become plasma cells. And these are really the two that I'd prefer that you know over the other one. Regulatory B cells, I don't want to get into too many details. They help regulate the response by secreting stuff, and like IL stands for interleukin, and I don't expect you to know that. That's a little too detailed. So what I do want you to know is that it's going to cause differentiation of different types of B cells, like memory cells and plasma cells. The memory cells, hopefully that's an easy one to remember because it results in memory. So our immune system remembers this infection, and it can mount an immune response against it more quickly later. Then the other type of cell that it will induce is plasma cells, and these plasma cells are the ones that secrete antibodies. And so you can see these antibodies are actually not in the shape of a Y technically. We have multiple Y shapes that come together in the center. This is still an antibody. Some antibodies are shaped that way. This is most likely, this would be, I'm assuming, an IgM antibody, but the text hasn't gone over that with you yet. But again, it is kind of Y shapes, but they're kind of all connected in the same spot in the center. So that's why it's generically shaped as a Y. Alright, so now that an antibody has been formed by those plasma cells, what do? I don't want you to know all the details for the exam, so you don't need to memorize this. All I want you to know for the exam is once those plasma cells are made, they can do various functions that promotes the immune system response. Now, if we want to get in the details, those antibodies can do different things called opsonization, neutralization, agglutination, lysing, and serve as antitoxins. I don't expect you to know the specific definitions of each, but they all serve a function in eliminating a pathogen. So opsonization, for example, is where antibodies coat the surface of a microbe, and that helps phagocytes like macrophages get a better grip on them so they can get them engulfed. So we're seeing that here. Here's our bacteria. It's been opsonized by various antibodies, and now it's being engulfed by our macrophage. Neutralization is where we will have an antibody, a collection of antibodies will fill the surface receptors on a virus, or any kind of active site on a microbial enzyme to prevent it from attaching normally. So if we look at, for the sake of viruses, the way that they normally would infect a human is to bind to a receptor on a human cell and get inside of it. But if these antibodies have now bound to all the receptors that are on that virus, it cannot bind to the human cell receptors and therefore can't get inside of the human cell. So we have neutralized the virus. Agglutination is the ability of our antibodies to attach to antigens of various pathogens and then cross-link together to form a clump. So that's what we're seeing here. We'll talk about that more later. Lysing is when a cell ruptures, so certain antibodies can result in the creation of little holes inside of a bacterial cell, which is going to cause it to rupture. And antitoxins, these are antibodies that are going to be specific to toxins that certain bacteria secrete. So if the antibodies are attached to the toxin can no longer do its job, which is something that would make us sick. So again, you don't need to know those for the exam, but just for demonstration purposes, very briefly, that's the various things that these antibodies can do. So next, let's look at the different classes of antibodies. This is the one that we typically see at first, our standard Y-shaped immunoglobulin or antibody. That's the shape of an IgG antibody. We also have IgD and IgE that are shaped as Ys. They have different functions, they have different sizes, and that's listed here in this chart. So I would expect that you should know, I don't expect you to know the half-life. Actually, I changed my mind, yes I do, because that's going to be important. You don't need to know these specific numbers though. What I want you to notice though is that IgG stays in the system longer than any of the other ones. So the half-life in the serum is essentially, in simple terms, is how long it's going to stay in your body. So the IgG antibody will stay longer than any of the others. Here's something else that's really important, especially going into nursing. The IgG antibodies can cross the placenta. None of the others can. And then finally, the biological function is listed last. So this IgG complex is produced by those plasma cells or B cells in response to any kind of stimulation, and also it's going to help by using memory cells responding to the second time of any kind of antigenic stimulus. So this is basically the most common type of antibody that circulates in our system, and it can do all kinds of different things that we talked about in that previous slide where we saw the opsonization, neutralization of toxins, and so on. Some classes of immunoglobulins are IgAs. So they are still Y shapes, but they're dimers, two of them that are butted up next to one another. Notice that it has a secretory component here. You'll also want to know that the dimer is secreted, hence why it has a secretory component, on mucous membranes. Something to think about. So if you have an upper respiratory tract infection, you're probably going to see a lot of IgA antibodies because it's an infection of the mucous membranes that line the respiratory tract. Here we see IgM. That one's going to be important too. It's a pentamer because it's made up of 5 monomers, five Ys. This is the first type of antibody that is produced in response to an antigen, and it can also serve as a B cell receptor. Our IgD is the receptors on B cells and a triggering molecule for B cell activation. And finally, IgEs, they are antibodies of allergy. Alright, so if you look at this table in the book, you'll see that it has a lot more to it. I eliminated the rows that I don't want you to know. These are the ones I want you to know. So you need to know these, but the only exception is you don't need to know the specific numbers here. You need to know that it's highest in IgG. That's what I want you to know. Then for the rest of them, know their functions. Know that IgG is the only one that can cross the placenta. Alright, so one way that we can track infection is by monitoring antibody production, because the more antibodies produce, the more antigen there typically is. And what we can do to measure that is measure what we call a titer. It's the level of antibodies in the serum over time. And it's a well-accepted principle that the memory B and T cells are only created from clones activated by a specific antigen. But new research has suggested that exposure to an antigen can result in memory cells that are chemically related to it, even if the antigens have not been seen by the host, which is really interesting. Especially in certain types of infections that constantly have different strains, like the flu, of course. Every time we turn around, there's different strains of the flu. So what this essentially is saying then is that if you contract the flu, you may be more protected from other strains of the flu that are chemically related to it. Same with getting the vaccine. It's like just because you get the vaccine, you may still contract the flu virus, but it will either not make you sick or not make you as sick, even if it's not the exact same strain that was in the vaccine, because it's chemically related to it. So it still serves some kind of component of protection. All right. The activation of specific memory occurs via our recognition of epitopes, and it may lead to the use of non-pathogenic microbes in vaccines against more dangerous ones. So again, that's pretty interesting with that new research that has come out. So let's look at our antigen responses. So what this is showing you is our primary and secondary responses to some kind of antigen or a pathogen. And it's showing you that in relation to antibody titer. So antibody titer, again, is concentration. When we are first exposed to some kind of pathogen or antigen, then there's going to be this period of time where we don't see a whole lot of increase, but it's kind of like our body, like when it first comes into contact with it, it's like, having to do all these things and figure out what this antigen is and get ready to mount this immune response. So there's a little bit of a latent period here before we see huge amounts of antibody titers come into play. And that's this blue line, or I guess I shouldn't say blue because that's blue. I guess that's a dark blue, or that we'll say it's purple. This purple line is representing the antibody titer of all IgM and IgG antibodies. IgM and IgG antibodies are the two that are standardly going to be detected via an antibody titer. And again, we said IgM is going to remain in your system longer. So if it also shows it separated out, we'll see that our first increase is going to be IgM in red, and then afterwards we see IgG. Now, this is going to show here a variable time interval, and all that means is the time in between the time you first saw the infection to the time you saw it the second time. So in your second round of coming in contact with the same antigen, your immune response happens very quickly. So to compare, we don't see that latent period in our secondary response, and we see this huge immune response that generates huge antibody titers very quickly compared to the first response. So looking here, comparing our purple lines, we see a huge difference, right? We see the antibody titer is much higher very quickly, like immediately, compared to over here we had that latent period. And then it's separated out again, IgM in the IgM in the red line and IgG in the blue. So again, we're going to have this huge increase of immune reactions via antibody titer in the secondary response versus the primary response. And that's actually, I'll go ahead and say this now, that's actually the foundation of vaccines. We'll talk about those later on today. But if you are vaccinated with, let's say, the chickenpox vaccine, that's your first exposure because it has antigens for the viral components of the chickenpox virus. And so you will have an immune response. It doesn't make you sick because we've eliminated the parts of the virus, or we've changed it in some way or form that it won't cause you virulence. It won't cause you to be ill, but you'll still mount an immune response. So then the second time, if you were to come in contact with the chickenpox virus, you have such a huge immune system response that even though you came in contact with it a second time, you won't even know that you got sick. Now, or you weren't ever sick. You won't ever notice it typically. Now, on the contrary, if you were to contract the actual chickenpox virus, you'll still mount an immune response, but you'll get sick from it. You'll be itchy, have all the bumps, fun stuff. And then the second time, you will not have an immune response. All right. So let's look at our next section, learning outcomes for section 13.6. We're going to list and describe our 4 different immune system states. We'll discuss the qualities of an effective vaccine. We'll name the 2 major categories of vaccines, talk about some subcategories, and we'll explain what herd immunity is and the risks that unfold when it's not maintained. So beginning with the different states of immunity. So acquired immunity, which is what we've been talking about all chapter, the adaptive immune system, can be acquired either actively or passively. Actively means you are exposed to antigen. And passively means that you are given antibodies. Both of those types of immunity can then be subdivided into natural and artificial means. So, natural active immunity is where we are naturally exposed to the antigen. So basically, in this type of acquired immunity, it's where we actually get sick from something. We get sick, pathogen enters our body, we make antibodies for it. We produce the antibodies. By the way, that's key there too. In active immunity, we are exposed to antigen when we develop antibodies. In artificial active immunity, we are not going to get sick, but we're still going to develop antibodies due to vaccination. All right, so vaccination. In passive immunity, we're given antibodies. So note that difference here, passive and active. In passive, we're given antibodies. Active, we're given antigen. And then that passive immunity can also be subdivided into natural and artificial, just like active was. In our natural passive immunity, our antibodies will be given naturally through breast milk, essentially from mom, because there are lots of antibodies and lots of other components that are found in breast milk that will boost the infant's immune system, such as this. And then in the artificial passive immunity, we're giving antibodies in a non-natural way. So, for example, giving someone antibodies to help them fight an infection, and such as gamma globulin, that's an antibody. So here we see some pictures to go along with that. Our active natural immunity, where we get sick. Passive natural immunity, where we are passing our immunity via antibodies through breast milk. Artificial immunity, where we can do it actively via vaccination, or passive immunity, which is artificial if we are giving a patient antibodies. Now let's talk about that artificial passive immunization where we're giving a patient antibodies. Again, antibodies are immunoglobulins, and we extract them from patients who are human donors. They've contracted whatever pathogen of interest, and they've made antibodies for it. We're going to take some of their blood and concentrate those antibodies and then give it to a patient that is currently trying to fight off that disease. Some examples where this is used is in Hep B and HIV. Also with COVID-19. So this is a procedure called immunotherapy, but this was something that started for COVID-19 in 2020. In the early days of COVID, during the pandemic, we did not have a vaccine yet. So one of the things that came out prior to the vaccine was the option of passive immunotherapy. And I remember even some of my friends had got this done and they had no idea about it. So it's kind of cool that it became such a more common thing that the public knew about than they did prior. So what that would happen is they would take this convalescent serum, which is the serum that contains the antibodies from people who were currently sick with COVID, and then recovered, and then given it to the people that were currently fighting it, having a hard time. So this approach is really useful, especially when there's no vaccine as of yet, which at that time there wasn't. So let's talk about vaccines. They have a pretty interesting history. So they were first recorded to be, the first recorded attempt of immunization occurred in the 6th century in China. And what they did was they dried up and ground smallpox scabs, and then they would blow them with a straw into the nostrils of family members that were vulnerable to the infection. Lovely. This led to other examples of this throughout history. It's called variolation. For example, in the 10th century, we would see deliberate inoculation of dried pus from smallpox pustules, and then it would be basically inoculated into the patient's arm of a healthy person So in a way, it's still it's exposing them to the infection. This was a common practice that was used in various parts of the Far East. It was brought to England in 1721. Unfortunately, this wasn't like the most ideal method of vaccination, but it was the best they had at the time, and it was better than nothing for them. So vaccines are not anything new necessarily. This is, again, first attempt at trying to immunize individuals from infection. Dates back all the way to 6th century China. But again, it did come at costs. There are still people that did die of smallpox or whatever else they tried to vaccinate, quote unquote, people from back then. Many of their recipients and their contacts still died, just not as many that normally would have. Now, jumping forward to 1796, famous Edward Jenner. He was inspired by a woman, and she was a dairymaid, and she had been infected by cowpox, and he noticed that she was immune to smallpox. And he thought, is there a relationship here? So he tested this theory, and how he tested it is pretty, highly unethical, but hey, this was the 1796. I guess they didn't have laws like this back then. But what he did was he injected a young boy with material from the human cowpox lesions of a person that had cowpox. So he allowed this boy to get sick with the cowpox, which is less fatal than smallpox, and then exposed him a few months later to smallpox, and the boy was immune. Very unethical of him to do that. Would have gotten a lot of trouble nowadays. So this use of this vaccine was first regarded with fear, though, and skepticism, but now has been later adopted once it now has been proved successful since 1796. So that's where we get our modern day foundations anyway in vaccination. Obviously, we don't expose people to cowpox to get them immunized to smallpox nowadays. This is just where it all began. All right. But let me back up here, and again, the foundation is that we learned that immunity could be attained with inoculation by selected antigens. But the way they have done that has changed drastically since 1796 or the 5th century China. But this is our brand of acquired immunity that was the artificial active state. And the basic principles behind vaccination is that we are going to stimulate a primary immune response and a memory response, and thus create lots of antibodies, high titers for this particular pathogen that we will then elicit a huge immune response to if we come in contact with it a second time. So that future exposure will be basically like nothing. Last point, vaccines have profoundly reduced the prevalence and impact of many infectious diseases that were once common and deadly. Let's look at types of vaccines. So the first type we'll describe are whole cell vaccines. And I don't really like this term because whole cell vaccines have subcategories that include killed vaccines and inactivated vaccines. And the killed vaccines are prepared using whatever the desired strain or strains of bacteria are that we are trying to make a vaccine for, and they're called the killed vaccines because we are going to treat those cells with chemicals or radiation or heat or any kind of treatment that will destroy the cell's ability to make someone sick. That would be like the virulence, but it doesn't destroy the antigenicity. So that means that a killed vaccine is going to contain cells, which could be bacteria, etc., protists, fungi, but they've been treated with certain things that will cause that cell to no longer be alive. But it still can produce antigenicity, meaning even though it's dead, our body can still detect it. So that means then we're not going to get sick from it because it's dead, but our body will still be able to recognize it and create an immune response and be prepared for it the next time it comes in contact with it. Now, again, the reason why I don't like the word whole cell vaccines is because whole cell vaccines also is going to incorporate inactivated vaccines, which are specific to viruses. And viruses are not cells, so that's just my little personal opinion, I suppose. But killed vaccines are going to be related to whole cell vaccines that are for bacteria, and an activated vaccine is going to contain a virus that has been inactivated, which means that it will again be exposed to some kind of chemical or radiation that will not, you can't say it's going to kill it because it can't be, viruses aren't alive, so you can't really kill them, but it inactivates the virus where it no longer makes you sick, but it still provides our immune system with an antigen for a response. All right, so next we have live attenuated vaccines, and these are going to contain microbes where the microbe is still alive, but its virulence has been attenuated, which means lessened. So you're giving the host the microbe of that you're interested in, and it's alive, but it doesn't have the ability to make you sick. So that's typically done by growing the microbe of interest in conditions that it's not used to, which will cause it to mutate, and eventually, and it will eliminate some of the viral or virulence factors. And some examples of vaccines that have been used with this method are the MMR, the measles, mumps, rubella vaccine, as well as the polio vaccine. All right, so let's look at those two contrasted next to one another. These are both the whole microbes. They stimulate immunity but cause no disease. Here's the killed cell or inactivated virus type. So we have our pathogen, which could be, a bacteria or a virus, whatever, but we're going to expose it to heat. And so in this case, with the cell, it's dead. But antigenicity is retained, meaning it still has these antigens on its cell membrane that can stimulate a response if we administer it into a human. Likewise, here we have our virus. It's technically not dead, it's inactivated, but it can still cause an immune response. So it doesn't make you sick because it's inactivated or dead, but it can still mount an immune response. Our live attenuated cells or viruses are going to be reduced in their virulence ability. So virulence is eliminated or reduced. These cells, or let me say this cell, is still alive. This virus is still active, but it has antigenicity without virulence. We administer it to a patient, they get an immune response from it, they come in contact with it a second time, they won't get sick. Another type of vaccine are subunit vaccines. Subunit vaccines are kind of just what they sound like. It's a subunit of a pathogen. So if we know the exact epitopes, the region of antigen that stimulate immunity, if it's known, we can produce a vaccine called a subunit vaccine. So if we have a virus here and we know that this specific thing, this part of it right here, is what our body detects, then we can harvest that, duplicate it, and administer it to a patient. And instead of it giving them the whole virus for our body to deal with, now we're just giving it that specific part that is the epitope that our body recognizes. So again, in that sense, there's no way that vaccine can make you sick, but it still provides antigenicity. Same concept with bacteria. Here we have a bacteria cell. You'll see there's certain components. These are antigens that can stimulate immunity. We'll remove those antigens from the bacteria and make a vaccine with just those antigens instead of the whole bacteria cell. Lastly, we'll talk about DNA viruses. There are a few other DNA vaccines. There are a couple other vaccines that are discussed in the text, but we're not going to cover those. So the only ones I want you to know, as per usual, are the ones we talk about in these lectures. Our DNA vaccines, we're going to have a DNA that is in a microbe, and it is inserted into a plasmid vector and then inoculated into a recipient. So at that point, then our human cells can take up this plasmid and express that microbial DNA in the form of proteins. Now, these foreign proteins will be recognized by the immune system and cause that immune system response that will result in B and T cells and memory cells to be formed. And so, to show you a picture of what that looks like, here we have our hypothetical bacterial pathogen. You see that it has a genome in here, its chromosome, and if we know that this region here will generate a protein that our body can recognize, we can cut that out. And there's ways that we can do that we won't talk about here, but some of you biology people may remember us talking about restriction enzymes. So here's another application. But we can cut this little chunk out and insert it into a plasmid. And a plasmid is a ring of accessory DNA in bacteria that we talked about way on back several chapters ago. So now what we're seeing here is we fuse this plasmid to this DNA of interest. We can put in a vaccine, inject it into a subject, and now these human cells can accept that plasmid. It's going into the human cell, and so now what we see is that this human cell will then begin to transcribe and translate whatever genes are on that plasmid, and those genes on that plasmid are some kind of bacterial antigen, and so now it will express those antigens on the host cell surface to serve as a flag essentially for the immune system that says, hey, I'm trying to stimulate a response here. I've got this protein. It's not human. Come take a look at it. And then things will move on from there as described in our immune response. Now, how can vaccines be given? Many of them are administered via the following routes. We have subcutaneous, which means under the skin, or intramuscular, which means into the muscle, or intradermal, which is into the skin. But others can also be given in nasal and oral forms. That's not as common, but these will stimulate IgA production on the mucous membranes. They're easier to give than injections. They're more readily accepted since it's not going into your body for some people, I suppose, for whatever reason. But so there are different routes of administration, but typically we're looking at those first three. All right, so I think that's everything for this chapter. Good luck on your upcoming exam once you get done with chapter 14, and I hope you have a great weekend, and I will see y'all again, or quote unquote, we'll talk again soon.